In the previous lectures, we considered what happens to allele and genotype frequencies in a large random mating population with no selection, mutation, or migration. We saw that after the population reaches Hardy-Weinberg equilibrium, the allele frequencies remain unaltered from one generation to the next, and the genotype frequencies are functions of allele frequencies. In other words, we developed a null model for the behavior of genes in the idealized populations. Now it's time to explore how allele and genotype frequencies behave in real populations. I'm Mohammad Sahebhonar, and for the next remaining lectures, I want to explain how populations evolve when the assumptions of Hardy-Weinberg law are violated. Formerly, we assumed a large population. However, in reality, populations are not large. In this video, I want to consider what will happen across generations to the genetic constitution of populations when this assumption is violated.
Consider one large population with no selection, mutation, or migration, in which mating is random, becomes divided into 20 small subpopulations. Each subpopulation is mated randomly for 100 generations. For the first scenario, there are 10 males and 10 females, 20 individuals in each population. In this simulation, we are focusing on a single locus with two alleles, A1 and A2. Imagine that in the present generation's gene pool, allele A1 is at frequency 0.5. For now, I want to focus on theoretical consequences of finite or small population size on the genetic constitution of successive generations. As depicted in the graph, each line tracks A1 allele frequency in each population. The allele frequency in every population has changed in an erratic manner from generation to generation, with no tendency to return to its original value. The direction of changes is unpredictable. In some populations, the frequency of A1 drifted to 1 and A1 became fixed. In other populations, A1 reached a frequency of 0 and disappeared from those populations altogether. In a few populations, A1 is still segregating. Let's rerun the simulation several times and see what will happen to the subpopulations. In the large population, the probability of drawing a gamete that contains a specific allele is equal to the corresponding allele frequency. Now imagine an idealized population that is finite in size. In this case, the probability of drawing a gamete that contains the specific allele is no longer necessarily equal to the frequency of that allele. By chance, some individuals pass more alleles to the next generation than others, and that may change allele frequency from one generation to the next. This mechanism of allele frequency change is called random genetic drift. Because of the random change of allele frequency, every population evolves independently, which leads to genetic differentiation among populations. However, within each population, genetic variation reduces from one generation to the next, and individuals become more and more uniform in genotype. Technically, inside populations, homozygosity increases and heterozygosity decreases. The increase in the frequency of homozygous genotypes is the basis for the loss of fitness that results from inbreeding. Genetic drift has a more rapid and dramatic effect on allele frequencies in small populations than in large populations. To show that, let's simulate 50 individuals in every population. Based on the result, day 1 is segregating in almost half of the subpopulations in the last generation. Now, let's increase the population size to 100. In this case, more subpopulations are segregating in the last generation. Next, let's simulate 20 populations for 500 instead of 100 generations and 100 individuals in all populations. So given sufficient time, genetic drift can produce changes in allele frequencies even in very large populations. Finally, let's focus on the initial allele frequency. First, I want to raise the number of populations to 100 and run the simulation. Let's rerun the simulation for a number of times. There is a fairly equal number of populations drift to fixation or loss. I want to change the value from 0.5 to 0.8. This time, 64 out of 100 populations reached fixation. I prefer to rerun the simulation a few more times. Almost 70% of the subpopulations drifted to fixation. Now I want to choose 0.2 for the initial allele frequency. The key takeaway is that the probability of fixation also depends on the initial frequency of the allele. In sum, genetic drift is a powerful evolutionary mechanism in finite populations, results from violation of the large population size of Hardy-Weinberg assumptions. Genetic drift causes a differential reproductive success that happens due to chance. Only chance determines which allele produces more offspring. It's noteworthy that selection is another mechanism that affects reproductive success. However, selection acts based on fitness.